So last time we were discussing genetics. So can you tell me what is a dominant allele and recessive allele?
So a dominant allele is so uh, so a dominant allele is so it's the type of allele where only one copy needs to be present.
It is the basically that allele which shows the characteristics in the organism.
Yeah.
Okay, I
So
told you
dominant.
like yeah, the dominant means like if you have your father is having blue eyes and your mother is having brown eyes, and you you basically will be having the alleles of both the genes you are going to have. You are going to have from your father also, you are going to have from your mother also. But since you are showing the color of brown eyes, that we are showing the color of your mother's eyes, that is a dominant allele. You understood?
Yeah.
Yeah, whatever comes in the offspring, that is the dominant allele. So the recessive one is also present, but it is not visible. Okay, and dominant allele we always write it with the capital letter, and recessive allele we write it with the small letter. This is all we have covered last time, right?
Hello.
Hello.
Yeah. Could
Yeah.
you repeat that? It broke up.
Yeah, so we had last time we had covered that what is a dominant allele and recessive allele, right?
Yeah.
Yeah, and what is basically the Puneet square? Can you tell me that why it is required?
Puneet square.
Why
So
we why do we require a Puneet square? I told you we used to make a square. Remember?
yeah. So to show which
Yes.
uh, I think allele is like.
Basically, it is help to show the genetic cross. Okay,
Oh
which
yeah.
all alleles will be present in a genetic cross. So,
Yeah.
like, if I will give you an example again, if your mother has brown eyes and your father is having blue eyes, so then you can see what are the chances of coming to of the blue color or the brown color eyes coming into the offsprings, right?
Yeah.
Yes. So, is it clear to you? Then we
Yeah.
spoke about two inherited disorders, if you remember. Can you name those two disorders?
I think.
Did you
Um.
study? Did you study what I taught you that day?
Yeah.
Yeah, okay. So, which two the disorders, inherited disorders we discussed?
So I think one of them was um trying to remember
Okay, one was the cystic
poly,
fibrosis. Yes?
yeah, cystic fibrosis and
Excellent.
polylactic
Yeah, I'll tell you polytactile, right?
dactyl, yeah.
Yeah, so now what was cystic fibrosis?
So cystic fibrosis I think was when um
When a thick, sticky mucus was produced in the air passages and the person is not able to breathe, right?
yeah.
I
So
explained you the yeah, carrier concept also. What are the chances of inheriting cystic fibrosis? Do you remember that?
yeah.
Okay, and what about the this we have already covered. I'm just giving you a brief intro what we have covered right now. Okay, so the second thing we discussed was polydactyl. So what was polydactyl?
So polydactyl. So so it's when it has more than 5 fingers or toes
More
on
than
each
five.
hand.
Okay, an extra finger, or you can say yes, it is there. So we call it as that condition, we call it as polydactyl, right? Yeah,
Yeah.
and can you tell me if you remember cystic fibrosis, it was caused by the recessive allele, and
Yeah.
polydactyl was because
The
of the
dominant.
dominant allele. Remember, we made the crosses also. Do you remember everything? So
Yeah, the
I should not
capital,
repeat that.
capital C and small c.
Yeah. So I should not repeat that, right? You are clear about
Yeah.
it. Okay. So then this was the first thing which we discussed. The second we spoke about the inherited disorder last time, and the third thing was the regarding selective breeding. Now, what was selective breeding?
Selective breeding is when you specifically breed for a particular purpose of a breed,
Okay.
like a sheep. If a sheep has more wool and you want to breed that particular sheep, you would selectively breed that particular sheep.
Excellent. So, what is the disadvantage of selective breeding then?
So, so disadvantage of selectively. So, firstly, it can lead to like loss of species, like biodiversity of that particular species.
Okay.
So,
Excellent.
it can lead to loss of species variety, as well as it it can create offsprings with different traits.
Okay, excellent. Yeah, that's the basic disadvantage of the selective breeding. So now let's talk about the genetic engineering. So this was the first thing which we studied. The second was this, third was this. Now the fourth topic we are going to discuss is genetic engineering. So now, are you aware of it? The word genetic engineering?
No.
No, I'll tell you. So now what happens is, see, now let's talk about a farmer is there and he wants to selectively breed plants and animals. Okay, so now what is he going to do? How will he do selective breeding? Suppose of plants he wants to do, how will he do selective breeding?
So
First of all, he is going to choose the
is
organism's genes,
yeah.
right? Which thing has
Then
to be, yes, breed, right? So he is going
yeah,
to choose the organism's genes.
yeah.
Then what is he going to do? He's going
Um
to put that genes in that plant, okay? And then,
yeah,
yeah. So but if you see, this process is quite slow. Okay,
yeah.
whenever I'll explain you what is genetic engineering. But before that, I would like to tell you that like if you talk about a farmer, if he wants to produce a plant, especially through the selective breeding, now that organism, for the first thing farmer is going to do is that he's going to take the organism's genes, whatever has to be inserted to the other one. So when it takes that, so then what is going to do, then is to prepare everything for the selective breeding, what all things are required. But if you see, this is a very slow process and it will take at least generations to do that, right? Selective breeding, like you take two things and you breed them and then you make a product and you are not sure whether it is going to survive or not. So that thing is basically, it is going to take ages, right? Generations.
Yeah.
So now what happens is these scientists, they're able to produce, you can say, or you can say they're going to alter the organism's genes to produce an organism with a desirable characteristics. Like suppose we have two organism. One organism, uh, I feel the genes are not like that organism should have, you can say, a specific characteristic. So now what I am going to do, I am going to take that specific character from one more organism and then I'm going to produce an organism which should have that characteristic. Like I have two plants, or I have one plant, and I love its white flowers. So I want that in my plant only white plant should come. So I'm going to take that white plant gene, and I'm going to produce one more plant which uh, which will have white colored flowers only. So now this thing, this is through the genetic engineering only. So now if you see, the scientists, they are able to produce some plants which are resistant to the diseases also. Okay, you understood?
Yeah.
Yeah. So now if like some plants, they're having lots of diseases, so now what is scientists doing? They are making certain plants which are resistant to the diseases. So now this,
Okay.
we call it as this is called genetic engineering, or you can also call it as genetic modification. You understood? It is being modified. The genes are being modified with the desirable characteristic. What do you mean by the desirable characteristic? Like if I want white flowers, if I'm giving an example, that means that I want the white flowers only in the other plant, right?
Okay.
So now can you
Yeah.
tell me what will be the advantage of genetically engineered plants? We can increase the yield of the crop also. So, right?
Yeah.
And we can generate certain plants which are resistant to diseases. Not in fact plants only, we can make animals also. Is it clear to
Okay.
you?
Yeah.
Yeah, like I'll give you an example. We have cotton. Okay, if you talk about cotton, the example, through genetical engineering or genetic modification, the cotton is being produced to have high yields. High yields are there in cotton. Okay, we have corn. Corn has started producing toxins that can kill the insects if they come on that. Okay, and through genetic engineering we are able to make bacteria which can make lots of drugs.
Yeah.
Okay, which can make lots of drugs. So that's been made, so we call it as genetically engineered. So basically these genetically engineered organisms, they are made by the scientists so as to make sure that they have the desirable characteristic, whatever the desirable characteristic they want in that particular organism. So now can you tell me how can we alter the organism's genes? I'll change the slide.
Yeah.
So now can you tell me how can we alter the organism's genes? How can you alter the organism's genes?
So in the nucleus, the genetic material is found.
In the
You could al-
nucleus, okay,
you could alter the genetic material
okay,
by maybe adding an extra chromosome.
okay. So but like if you're talking about the genetic engineering, so how will you do that? See now, if
Um.
you want to create an organism which should have the desirable characteristics, the first thing is the scientist is supposed to take the genes from the organism, right?
Okay. Yeah.
Yeah. So the scientist is going to take the genes from the organism that shows the specific characteristic, the desirable characteristic, right?
Yeah.
Now once the gene is being taken, so what will happen? Now first of all thing, these genes which are taken from the organism, these genes we call them as foreign genes.
Oh, okay.
You understood the word foreign? It is being taken from outside. That is, we call
Okay.
it as foreign genes. So now what is going to happen is, these foreign genes, they are put into a plant, you can say. You put them into the, this foreign genes, you are going to put into plants, or you can put it at the animal cells, plant cell or animal cells, at very early stage of their development. You understood? When they're quite small, when they're cells, when they're just about to grow. So these foreign genes, they are going to put it in the plants or the animal cell. So then what will happen? Now if you see, as the organism, it develops, what is going to happen?
The
It will start
develop.
showing, it will show the characteristics, right, of that foreign
Yeah.
gene.
Okay.
Now this is how you can alter the organism genes. Is it clear to you?
Yeah.
So basically, how will you alter the genes? You are going to take, the scientist is going to take the genes from the organism, which you want the desirable characteristics. Now those genes, you call them as the foreign genes. You are going to put them into the plant or the animal cells. Then as the plant, and you will put it when it will be the early stages of the development of the plant or the animal cell. Then as the organism develops, it will start showing the characteristics. Like, let's talk about the disease resistant plants. So what is going to happen in disease resistant plants? The uh, the this, this special characteristic is being taken from a particular organism. Okay, that genes are being taken, disease resistant genes, they're going to be taken from one organism, and they're going to put it in the other organism, other plant cell. And that plant cell, eventually when it grows, that plant will be disease resistant. Clear?
Yeah.
So now we have some, if you have heard, the frost free tomatoes are there. The frost free tomatoes. Now what is that, you know?
No.
I'll tell you. Frost free tomatoes are basically, I'll explain you. There is a fish, we call it as the flounder. Flounder.
Okay.
Okay, that the name of the fish is flounder. Now flounder, it's kind of a fish which stays in a very cold water. So now that means it has got a gene which helps, or which prevents it for freezing, right? Now when something is there, the organisms which stay at a particular temperature, they acquire certain characteristics of that area, right?
Yeah.
Yeah. So now this fish, it is found in cold regions. So now flounder fish is found in the cold region. So that means it has got certain genes which help, or which prevents it to freeze. You understood?
Okay.
That
Yeah.
means it has got some anti-freeze chemicals, so that this fish can sustain in the cold water also. Is it clear to you? So
Yeah.
now the scientists, they have taken this, you can say, frost free, the frost resistance to this tomatoes. They have inserted the flounder's antifreeze gene into the cells of the tomato. Now this flounder, it produces antifreeze genes.
Okay.
Now these antifreeze genes, they are put into the cells
Tomato.
of the tomato.
Tomato.
Okay, cells
Okay.
of, I'll write it for you, cells of tomato. Now just a minute, tomato, this tomato. So now when it inserts into the cell of the tomato, what is going to form? So now this tomato which is being formed, it will it will no longer be destroyed by the freezing properties, by the
Oh,
frost, right? So that
yeah.
means it can be beneficial to the farmers also, for the farmers. So you should remember this example, that flounder is a fish which has anti-freeze genes because it lives in the cold water, and those genes are taken by the scientist and they are placed in the cells of the tomato. And when they are placed in the cells of the tomato, the tomatoes, they now have the anti-freeze genes. Now they are not going to get destroyed, and it is very beneficial for the farmers. So is it clear
Yeah.
to you? Can I change it? The slide?
Yes.
Yes.
Wait 1 second.
Okay.
Yeah.
So the next thing is, this was like, there's some tomatoes which are frost resistant. Let's talk about the bacteria. We call it as the genetically genetically engineered bacteria. Now, what is bacteria?
Um,
It's a
bacteria.
microorganism, right?
Yeah,
Yeah.
yeah. Some
So
bacteria are helpful for you.
okay, so see, these bacteria, they can be genetically engineered also. Like, uh, we can produce lots of chemicals from the bacteria. Okay, like lots of vaccines can be made, lots of antibodies can be made. Okay, so these genetically engineered bacteria can make lots of vaccines. The antibiotics can be made. You understood?
Yeah.
Yeah. So now if you see, the bacteria, they have a very important property, that they reproduce very fast. So now whatever chemical they are going to produce, so I can say that that chemical is going to multiply in a very short period of time. Now if you insert something in bacteria, okay, Rohan, if you put something in bacteria, what is going to happen? Hello?
Hello.
Yeah. So, can you hear me?
Yeah.
Yeah. So now, see, I told you that bacteria, it reproduces very fast. It has the property of reproduce reproduces very fast. Okay. So now, whatever you insert in bacteria, like if you put some chemicals in the bacteria, what is going to happen? The quantity of that chemicals will also keep on multiply as the bacteria keeps on reproducing. From one bacteria cell, it is going to make two bacteria cells. From two bacteria cells, they are going to make four. You understood? There is a bacteria, it reproduces, it splits up into two, then this will form 4. So again this will form 8. You understood?
Okay. Yeah.
Yeah, so this bacteria, not this bacteria, all the bacteria, they have a property that they reproduce very fast. Okay, so now if I insert a chemical here, this chemical is there. So now if you see, if the bacteria splits, you will be able to see this chemical here also. It will keep on multiplying. It makes identical copies of that. So if you see, this chemical now is been into the 4 cells.
Okay.
You understood now?
Yeah.
Yeah. So now that means it has the so if you modify, if you say genetically engineer the bacteria, so it is going to make the chemicals or vaccines or antibiotics very fast, because bacteria reproduces very quickly. So now if let's talk about our body, we have a we have a chemical in our body, and that chemical, let's talk about that chemical, we call it as the insulin. You know that about insulin?
Yeah,
Yes.
yeah, from
Yeah, so now what is the main function of insulin? It controls
uh
the blood sugar level, right?
okay, yeah,
Yeah, so basically it controls the blood sugar level. So some people, they do not produce enough of the insulin. Okay, so
okay,
that means they must inject it daily. If you are not able to produce so much of insulin in the body, so you must have heard people saying that okay, the people are taking injections of insulin.
yeah,
Yes, so now this insulin, they need how it, now this insulin, we are talking about the injections of insulin. If a body, if in a body the natural insulin is not being prepared, then people, they have to take insulin injections so as to maintain their sugar level. So now how the scientists, they are making insulin so fast? They are making with the help of these genetically modified bacteria. Okay, now they are going to, you understood?
yeah,
Yeah, so now this insulin they need can be made very easily with the genetically engineered bacteria. How it is happening? See now the genes which says for the production of the insulin, they are basically the genes. Okay, the genes, they are inserted in the bacteria. You
okay,
understood? Inserted in bacteria. Then what happens when they are inserted in the bacteria? This bacteria, it multiplies means it reproduces very fast. And then what happens? It produces insulin.
okay,
Is it clear to you now?
yeah,
So now because the bacteria, it can multiply very fast, so it produces large amount of the insulin. So then what happens is the bacteria, they are being removed and the insulin is being left, so the scientists can use that insulin in for making antibiotics or vaccines. So now is it clear to you?
yeah.
Yeah. So now can you explain me about the genetic engineering, what we have discussed?
So
What is genetically engineering? What will you say?
the genetic engineering is the process.
Okay. I'm not asking for the exact definition, just but at least try to explain me what we have studied till now.
Okay, so it so it's where like you try to manipulate an organism's genes.
Excellent. Yes. Whatever desirable characteristics you want.
Yeah,
Okay. So
yeah.
now, what do you say? The selective breeding is better or genetically engineered engineering is better, according to
Um,
you? See, if you see, I told you selective breeding will take a lot of time.
yeah,
Yes, because you have to
yeah.
take, yeah, you have to breed them and all. But if you talk about the
Yeah.
genetically engineered,
You can make faster changes to organisms than selective breeding.
right? So I think it is a much better deal, right?
Yeah.
So now, can you, yeah, so can you tell me, explain what is frost resistant tomatoes? Can you explain me that? What I have told you? How will you get frost resistant tomatoes?
So you get host resistance tomato from the flounder fish.
Yes.
Um,
What is the
flounder
property of
fish.
this? Yes, flounder fish.
So flounder, flounder fish have a characteristic that allows them to survive in cold and freezing temperatures.
Okay.
So
So that
when the sun,
characteristic, what do you call it as? That it has anti-freeze genes.
yeah, antifreeze genes.
Yes. Yes.
and then those antifreeze genes.
That they are going to, these anti-freeze genes, they insert it into the tomatoes, right?
Yeah,
Yes, and to have the the this free, the genetically engineered tomato plants, right? And now, are you clear about the genetically engineered bacteria, because that is also important?
yeah.
Yeah. So now, what is the characteristic of the bacteria?
So like, so it allows.
It reproduces very fast, it
Yeah,
multiplies very fast. Okay, so
yeah,
that is
yeah,
why whatever you insert, or whatever chemical you will insert, you'll get the products very fast. So that is why it is used to make the vaccines or the antibiotics.
yeah.
Okay, so now let's talk about the word clone. Now, what do you know about clone?
Okay, so clones are uh, like a exact replica copy of
Okay,
a living person.
so clones are basically, yes, the clones, they are the exact copy, or you can say which is genetically identical to the parent also, we can say that also. Like if you see two birds which look alike, okay, suppose we see two black birds. Now, can we say that they are the clones of each other? Or if there are two
No,
bacteria, why they look alike?
because they're lookalikes, but a clone needs to behave the same way as an exact organism.
But the two birds, they're behaving same. How will you say the two organisms, they're behaving same? They look like. So basically, they are clones only.
Oh, yeah,
Okay. So
okay.
now basically, a clone is an organism which basically, which is genetically, you can say, identical to its parent. Yes, you can say that also, that it behaves like that also, right? You're right. So that means the person should have the same genes as its parents, and therefore will look like if you talk about the bacteria. Now, can you make out the difference between these two? No, these two are same, like these three are same. So the offsprings, they are identical to the parent cell, right?
Yeah,
Yes, so they therefore they look identical. So now when bacteria, it reproduces, they divide into half. So like you have a bacteria. Okay, this is the bacteria. I'll show you here. Now when they split, how do they split? They split up into, like, it is like this. The cytoplasm will start dividing, and then eventually the two same cells will be formed. So whatever is present here, it is been split up into two cells. Okay, and these two cells, they are exact copy of this bacterial cell. So now that means these are called clones.
yeah,
Okay, and see, if I was telling you an example of insulin, I told you that bacteria, it reproduces fast. So can I say that it is making clones? So that is why the insulin is also making clones.
yeah,
Bacteria, you understood, the bacteria is making clones, so the insulin is also made as the clone only, because it is going to make in that only, right?
yeah,
Now, if we talk about asexual reproduction, now bacteria, they do not require a partner to reproduce. Okay, so now only one parent can reproduce. One parent is required to reproduce. So that kind of reproduction, when only one organism is involved, we call it as sexual or asexual.
asexual.
Asexual. And now asexual reproduction, it happens with the cell division. This is what a cell is dividing. If you could see, that this cell, it is going to divide like this, and then two cells are making. You understood? Hello?
Hello?
Yeah,
Yes?
you understood?
Yeah.
Yeah, so now how is the asexual reproduction takes place? It takes place with only one partner. Only one partner is needed. Only one organism can reproduce. That we call it as asexual reproduction. No partner is required. So now if you see in this case only, the asexual reproduction, basically it happens when the cell starts dividing. Now if you see this cell, it has started dividing and it has formed the two offsprings. So now lot of plants are there which reproduce asexually.
Yeah.
Can you name few of them? Do you know?
Uh, I'm not sure.
What, you name any of them. If you see potato plants, if you see strawberry plants, daffodils, all these plants, they reproduce asexually only. If you talk about the potato plants, what is there in potato plants? There are tubers which are there, right? And when and
Yeah.
in each tuber the new plant comes out. Or if you talk about the strawberry plants, they have long stems like of these, and then they have the platelets are there. So now at the end, so what is it from here? From each point you can say a new strawberry will come. So all these plants, they are reproducing strawberry, the asexually. You understood?
Okay.
So now if you
Yeah.
talk about daffodils, now in daffodil what happens? A new bulb is being formed, and from that bulb a new flower comes out. So you understood now asexual reproduction. Now how can you make clones? Can you tell me how will you make clones? Have you ever tried making a clone?
No.
See, I'll give you an example. You must have done that also. When you want to grow, if you like gardening, Rohan, what do? You take a cutting of the plant from a particular plant, and then you take it and you grow that plant. You understood? You take a
Okay.
plant cutting and you make a clone. How will you make a clone? Now you know that certain plants, they can be grown from the cuttings also. You take a cutting, that means you cut a small section of the plant and then you plant it. Then what will happen? After few days you are going to see a grown up plant, a new plant from that cutting.
Okay.
And have you tried this?
No.
No, okay. So now, see, whichever, like you take a plant, you take a money plant or any plant you take a small cutting of that plant and you grow it, you plant that, and it will grow up into a new plant. So that means, and that plant is going to be the same as that of the parent plant, okay, from which the cutting has been
Okay.
taken. So that we say that you have made clones. Okay, sometimes what
Okay.
happens is you dip you take the cutting, okay, and you dip that cutting into a special chemical. We call it as the powder. It is called rooting powder. So that,
Okay.
yeah, to encourage basically the cutting to grow roots. When you dip it into the rooting powder, then the roots are going to come quickly. Now, if
Okay.
what is the advantage of this technique? Can you tell me cloning? Why we require cloning?
So that you can get the characteristics from an organism.
But then, what is the difference between genetically engineered the organisms? Also, you are also getting a new characteristics also. See, in cloning, you don't get a new characteristic. In fact, you get new plants very easily. You understood? You have taken a cutting and you have grown that plant. So that means if you see, the plant is going to grow up very quickly. and you don't require a lot of things for that, or the chemicals for that. It is a cheap process also. You take a cutting, you plant it, and the plant, you are going to get a new plant, right?
Yeah.
Yeah, and
Yeah.
if you see all the plants, they are genetically identical,
Okay.
so they are going to have the same characteristics,
Yeah.
right? These were the advantages. Okay, so now I'll change the slide.
Oh, 1 second. Yeah, now you can change the slide.
So now you must know what are the in cloning, what are the advantages of cloning. Now advantages we have already discussed, that you can say the new plants can be easily formed. Okay, the technique, this is very cheap. The plants, they are genetically identical, so that they can have the desirable characteristics. Now let's come to the disadvantages. What do you think, what can be the disadvantages?
Uh, Disadvantages could be like it could develop. Wait, I'm not really sure with cloning.
See, now cloning means that you're going to get the same variety of the genes, right?
But yeah,
So now
so
when you get the same variety of the genes, so the chances of getting the disease or will be more, which is going to destroy the
oh yeah,
species also, right?
yeah,
Or if there's any change in the environment of the plant, what will happen? The species can die.
yeah.
So this is the disadvantage of the. Now this we were talking about the plants. Okay, these advantages and disadvantages we were talking of the plants. Now let's talk about the animals. Okay, we have discussed the advantages and disadvantages of cloning in plants. If we talk about animals, what do you think? What can be the advantage?
Um,
See, now I'll give you example. In animals, we have endangered species, right, Rohan? Endangered species are there. So now all the species which are going to get extinct, if we try making clones of that, do you think we can protect them from getting extinct or getting endangered?
yes and no.
Yes and no. Why?
Well, yes, it will increase the population, but no, as it's because if it's the same, it will create diseases, and that one might not be as healthy and the perfect one to like to clone.
Yeah, I understand,
'Cause-
because you're saying that if they're getting extinct, that means they are getting extinct due to some change. That is whether it is a change in the environment or right. But still, we can preserve them. If you can do cloning, still you'll be able to preserve them. Okay.
Yeah.
Yeah, you are right. You are giving me a yes answer also, no answer also. But we can at least try to save those through cloning.
Okay.
Okay, so like I'll give you an example. If you know, if we talk about animals, we're talking about, there is a sheep named Dolly.
Oh
Okay,
yeah, Dolly.
yes,
Yeah.
Dolly. It is the first mammal actually to be cloned using the cell of the sheep.
Okay.
You remember?
Yeah.
Yes. So now I'm just giving you now, through this Dolly we can in fact whatever there is desirable characteristics which we want, we can do that also, right? We wanted a specific characteristic in the sheep, we can do that also. So now how will you do animal cloning? Let's discuss that first. For animal cloning, it has been animal cloning is done by two things. One thing is you clone the cell. We call it as cell cloning. Now in cell cloning, what we do is the scientists they clone the human cell in the laboratory, and then they can use it for the research, or you can say it to remove the diseases also. You understood? Now animal
Mhm.
cloning is what? The cell cloning. Our cell from the human body has been taken, and it has been used for the research so that they can try to remove the diseases. Clear? So
Yeah.
we have now tissue culture. Now what is tissue culture?
I'm not sure.
I'll tell you what is tissue. Tissue is basically when lots of cells they come together and they carry out one function, that we call it as a tissue. So now if you tissue culture means like the scientists then they can grow new skin, cartilage in a very in a laboratory, you can say, in a very sterile methods. And uh, and then what they can do, like suppose I'll give you an example. If there is a victim of burn, okay, so now the scientists hello.
Hello. Yes.
So now if you have a victim of a burned skin, you understood, a person is there whose skin is burned. Now he or she wants that okay, the burn should not be visible. So now the scientists they can, or the doctors you can say, they can make a tissue, kind of a skin tissue, and they can put that patch on the place of the burn,
Okay.
right? So now that
Yeah.
also is done by cloning only. Now how it is done? They are going to take the cell of or the tissue area, some part of their body only. From the body only they are going to take the, suppose one victim is there, a burn victim is there. Now doctors are going to take a tissue of that burn victim, and then it is going to grow up in the laboratory in a very, you can say, sterile method. Okay, full sterilization or full sanitization is taken care of. And then when it is being made, when it is being cloned, it is similar to the cells of it is similar to the tissue. So then it can be used to put it on the area of the burns of the victim. Is it clear to you now?
Yeah.
And then what is cell cloning? Cell cloning, the in the lab, if the cells are cloned, so the scientists they can use it for the research. Now can you now are you clear with this? So
Yes.
now can you tell me what are the advantages, disadvantages of both the cloning in plants and animals? Let's talk about plants. Can you repeat what I have told you?
Um. So the disadvantages in cloning is
No, I'm talking about first advantage of cloning of plants. Let's talk about plants. One.
the advantages of cloning of plants. So the they can be produced faster.
Okay.
Yeah, and like so the plants will improve in the sense that
Okay,
they
the
will,
desirable characteristics you can get.
yeah,
Yes, okay. And is this technique cheap or expensive?
it's cheap.
It's cheap. You take an example, you think in your mind the example of a cutting which you are going to take it from the plant. So it is so easy to make it also, right? And the
Yeah.
disadvantage is,
The disadvantages is they're prone to like diseases.
okay.
as
And
well as they may develop molds,
okay, excellent. This I have not
as
given you
well
the example, but you have given me the example. Very good.
as
Okay, and you can say there. Okay, there is a high chance of diseases, or if there is a change in the plant's environment, it is going to destroy the species also, right?
yeah.
Yes. So now what about the animal cloning? The first thing in animal cloning, it is done by two things, two ways you can say. The first one is the cell cloning, and the second one is the tissue culture. So are you clear about that?
Yeah.
And then let's talk about the advantage of animal clones.
So the advantages are that they offer great benefits. For example, a sheep, it allows farmers to accelerate the reproduction of most of their livestock in order to produce safe and healthy food.
Okay. And in fact, the endangered species will all also, they can be
Yeah,
cloned, right? So it will prevent them from the extinction.
yeah,
Yes. So now till now everything is clear to you.
yes.
So now let's just start with the next topic, that is the biotechnology. Okay, that's the yeah, biotechnology. It's a part of this only,
Okay,
biotechnology. There are lot of subtopics of this chapter. So now let's talk about the biotechnology. Now if you see, you make, what is biotechnology basically? In biotechnology, it is a field of the science in which deals with the study of the microorganisms, you can say. So now if you see, lots of food and drinks, you make it with the help of the microorganisms. For example, can you give me any microorganism which is required to make a fruit food?
yeah, okay.
Okay, give me. It's a very common example you have given me so many times. Yeast.
Oh, yeah,
Yes,
yeast.
we add yeast. Why we add yeast to the bread? So as to make it rise, right?
Yeah,
So now if we talk about biotechnology, biotechnology is basically the use of the biological processes or the organisms so as to create, you can say, the useful products. And most of those useful products, they are like food or drinks.
okay.
Understood? Through biotechnology, you may biotechnology is basically the use of the biological processes, and so as the organism can make up useful products. Now if we talk about the yeast, yeast is a microorganism, and that yeast, it it produces, it helps to produce bread.
Yeah,
Yes. Now what is yeast basically? Yeast is a microorganism. Okay, so you must know that what is yeast. Yeast is the microorganism that helps in the production of the bread. And does that reaction takes place anaerobic or aerobic?
anaerobic.
Anaerobic, because if you see the yeast, it basically, it takes place in the fermentation reaction. We just did in chemistry also.
Yeah.
Now what was fermentation reaction?
So fermentation reaction was when the microorganisms using anaerobic respiration, they
In fermentation, if you see the glucose, in the absence of oxygen, it gets transformed into ethanol
yeah, ethanol
plus
and
carbon dioxide
yeah,
plus releases energy also, right?
yeah,
So now what happens? So this yeast is basically the microorganism which is being used in the production of bread and mainly alcoholic drinks. So these products, they are useful for reaction fermentation, if you see. So now fermentation, you know that this yeast, it can respire without oxygen. So that is why it takes place in fermentation, because here there is absence of oxygen. So when there is
Yeah.
absence of the oxygen, the yeast, it can easily transform glucose into ethanol, carbon dioxide, and energy. So okay,
Okay.
so I'm just giving you an introduction of this thing. So this we won't be able to finish right now. We have biotechnology 1, biotechnology 2 topic, and the third one is enzymes. You understood? Now
Okay.
these three topics we can cover them next time whenever we meet. Because
Okay.
yeah, okay. So now about the yeast, I have given you introduction. So now let me tell you one more thing. Let's cover up the part, the bread. How do you make bread? So for making bread you require flour, water, and yeast, right? How to
Yeah.
make bread, I'll say. What do you require for that? You require flour,
Yeah.
water. This is a part of the biotechnology 1 topic only. Flour, water, and yeast. You require these three things. And then what happens? You make the dough,
Okay.
right? You make the dough and then you keep it in the warm place. And then what happens? When you keep it at the warm
And
place,
then it rises.
yes, so the yeast, it acts on the dough, and the carbon dioxide starts releasing, right? When it is
Yeah.
released, it causes bubbles and the dough, it rises up. So this dough is then baked. This dough is then baked, and then what do you get? The ethanol, it gets evaporated, okay? And then whatever carbon dioxide is there, it also helps the bread to rise up. You understood?
Yeah.
Yeah. So now, if I ask you, name the gas that makes dough rise, can you tell me?
Um. So the yeast makes the dough rise.
I'm asking, name the gas.
Oh. Carbon dioxide.
Carbon dioxide. So basically, you take these three things, ingredients, you mix them, you make a dough, you keep it at a warm place. This yeast is going to act on the flour, the flour basically. And what is going to happen? Whatever is present in the flour, whatever sugar is present in the flour, it is going to get converted to ethanol, carbon dioxide, and this carbon dioxide helps the dough to rise up, and ethanol, it evaporates. So now is it clear to you? And you get the bread which rises up.
Yeah.
Okay. Yeah. So now I'll just write few questions for you. You try to attempt that. Okay. So can I rub it?
Yeah.
Okay. The first thing you must know. I'll ask you next time. Okay. You must write it. What's the difference between the selective breeding and genetically engineered? Okay. You must tell me this, and you must know you must remember the Punnett squares of the inherited disorders which we know.
Yeah.
Okay. So that is you try to attempt this thing, and so you try these, and the rest of the portion we can cover next time. Biotechnology 1, biotechnology 2, and the enzymes. Enzymes are also very important. I want to take it as a nice topic, because if you see in that, you get to know lot of things. Okay, there is the word denaturing of enzymes that you must know. So I'm going to take up that also. Anything you want to ask me, Rohan?
No.
Sure, you have understood what we have I have taught you today. Is it clear to
Yeah,
you?
I have understood. Okay.